Welcome student in part 2 of chapter 13, Why Do We Fall Ill? Now let's discuss about the non-infectious diseases. These diseases may be of the following types. It could be deficiency disease, could be degenerative disease, could be cancer, allergic disease, metabolic disorder, or injury. Deficiency disease is caused due to lack of certain essential substances in our diet like protein, vitamins. An example of such diseases are scurvy and rickets. Degenerative diseases, these are caused due to degeneration of the tissue. As we turn old, our tissues get degenerated. One of our tissues, an example of such disease is cataract. Then the third type of non-infectious disease is cancer. It is caused due to the uncontrolled growth of tissue in any part of our body. Then the fourth type of non-infectious disease is allergic disease, which is caused due to hypersensitivity of our own immune system towards the foreign substances. An example is asthma. Then comes the metabolic disorder. These diseases are caused due to defects in our metabolic reaction. And the last one is injury, that also comes under non-infectious diseases, and that is caused due to injury and damage to any part of body by accidents. Infectious diseases. Infectious diseases are caused due to infectious agents. Disease that cause the disease causing organisms are called pathogens or infectious agents. Such are viruses, bacteria, fungi, protozoa, worms, etc. Now the question comes, why do we need to classify pathogens? Like, why do we need to know about infectious agents? It is required for a treatment, right? Treatment given to a particular category does not work for other categories. Like the treatment for bacteria is different than viruses. The treatment for viruses is different from fungi. So it is necessary to know the causative agent, infectious agent to treat. Right? So, however, the members belonging to a particular group, say bacteria, they have a similarity in their biochemical pathways, in their structure, and these are not shared by the members of other groups. So, a drug that blocks one of the biochemical pathway of one group may be effective against many members of the same group. Like here, example is given antibiotic penicillin. Penicillin inhibits the building of protective cell wall of the bacteria. As a result, the bacteria die because cell wall is not built. Since many bacteria use such processes to build cell walls, penicillin is effective against many such bacteria. Antibiotics, however, are not useful for killing viruses because they don't have their own pathway. They use the host pathway, right? So instead of that, our body secrete an antiviral protein which is known as interferon when we get infected with viruses that combat the virus for certain diseases. Here are some types of pathogen, description, and human disease that are caused by these pathogens. First one is bacteria. The example is Escherichia coli. This one is single cell organism without a nucleus. And the type of disease that they cause are strep throat, staphyl infections, tuberculosis, food poisoning, tetanus, pneumonia, syphilis. So these all are the bacterial diseases. These all are the bacterial disease. Then comes the viruses, which are thread-like particles that produced by taking over living cells. Example is given, herpes simplex is an example of a virus. The common diseases, so the common viral diseases are common cold, flu, genital herpes, cold sores, measles, AIDS, genital warts, chickenpox, and smallpox. The other type of pathogen is fungi. These are simple organisms including mushrooms and yeast. that grow as a single cell or thread-like filament. Here on our screen, we have death cap mushroom, and the diseases that are caused due to fungi are ringworm, athalate foot, tinea, candidiasis, histopalmosis, mushroom poisoning. The last type of pathogen is protozoa, and these are also single cell organism but with nucleus, and the disease is malaria, travel diarrhea, or sleeping sickness. Then the question comes, why do we need to understand the characteristic of this infectious agent? If we understand the characteristics, we can treat the disease caused by them in a better way. So we need to find out which medicine will work on which infectious agent. So for that, we need to understand the characteristic of all these infectious agents. Some common characteristics of infectious agents are like viruses always live inside the host cell. Bacteria do not generally live inside the host cell but inside the host body. Bacteria do not generally live inside the host cell, but they generally live inside the host body. Virus, bacteria, and fungi, they multiply quickly, so it is very important. If a bacterial, viral, or fungal infection is there, we need to stop their multiplication. Then worms do not multiply quickly because they are multicellular organisms. Here on our screen, we have how the penicillin helps in fighting against bacteria. Antibiotics are generally used to block the growth of bacteria in this way or another way. The bacterial cell, here in case of penicillin, cell wall synthesis is stopped, is inhibited by in presence of penicillin. So the bacterial cells that will grow in presence of penicillin will not be able to prepare its own cell wall. Penicillin is an antibiotic that prevents the growth of the cell wall and hence bacteria die easily. So it is used for fighting against different kinds of bacteria. Now, based on the means of spread of a disease, we can classify diseases into different categories like airborne disease, waterborne disease, foodborne disease, lifestyle diseases, vector-borne disease, and sexually transmitted disease. Airborne disease, the causes is air, food by consumption of contaminated food. Waterborne disease is by consumption of contaminated water. Lifestyle disease, it occurs because of the poor or unhealthy lifestyle. Vector-borne disease is caused due to animal that carries this infectious agent from sick person to another person. And sexually transmitted disease, it caused due to sexual contact from one person to another. Examples of airborne disease are tuberculosis, common cold. Foodborne is food poisoning, typhoid. Water is cholera. Lifestyle disease is diabetes. Vector-borne disease, malaria, dengue. And sexually transmitted disease is AIDS and syphilis. So here on screen, it's given that how the disease is transmitted from infected person to a healthy person through direct contact, by air, indirect contact, through food, via vectors like mosquito and insect, or rabid animals. Now the next question comes, where do the disease causing germs live in our body? There are number of tissues and organs in which in our body where these microbes can get attached to. Generally, the point of entry decides where these bacteria, these pathogens will go. For example, any microbe that enters through nose is likely to settle at the lungs. This can also be seen through a sign and symptoms of a disease, as only those organs and tissues seem affected where these microbes enter. But there are some other common diseases also that there is no tissue specific. These are not tissue specific. So in that case, the sign to observe is inflammation. Inflammation, our body has an immune system in which it creates a cell that fight against disease causing germs. And this process of recruiting cells to kill the infectious agent present in our body, and this entire process is known as inflammation. The inflammation process shows different effects on our body such as fever, swelling. Hence, we can say that likelihood we are going to be affected by disease is determined by our immune system. The effects here are some effects of inflammation: heat, redness, swelling, pain, and loss of function. These are the effects of inflammation. Heat, pain, heat of the particular body organ, redness of the particular body organ, swelling of the particular body organ or the tissue, pain in any part of our body, or the dysfunction, loss of function of the part of body. These all shows that likely we are affected, we are suffering from a kind of infection or disease. Now moving ahead with principles of treatment. The first thing is to diagnose, then comes a prevent, treatment, and finally it comes a cure of the disease. We may treat infectious disease in two ways. Number one is by killing that infectious agent inside our body, or by reducing the effect of the disease or reducing the symptoms. We can reduce a symptom in following way: by taking rest to conserve our energy so that immune system can fight against the disease; by taking medicine to reduce the common symptoms such as fever or pain and hence reducing the disease. So to reduce the symptom, there are two ways: by taking rest or by taking medicines against it. We can kill the infectious agents in our body in the following way: by taking medicine, those are antibiotics against the bacteria, or antiviral against the virus, or antifungal medicines that function against fungus infections. So these are the way that which we can kill the infectious agents or reduce the symptoms of infections. But antibiotics are ineffective against viruses. Why? Because antibiotics commonly work by blocking the biochemical pathway that are important for bacteria. Whereas viruses, they don't have their own metabolic pathway. They don't have their own cell. They utilize host body to function. Right? That's why when antibiotic is being taken, the antibiotic will hamper the metabolism of bacteria and kill them. And antibiotics do not work against the virus because viruses do not use such biochemical pathway. They don't have their own machinery. They use host cell machinery for making proteins. So the most effective way to treat viral infection and disease is through vaccination, as it can prevent a person from getting the diseases in the first place. So antibiotic doesn't work against viruses. So for that case, to fight against a viral infection, vaccination is important. In principles of prevention, are there any limitation of treating infectious agent via medicines? Yes, there are three limitations while we are treating infectious agent. Our body function might not be able to recover easily. If a person is not recovering easily, that may be the limitation of treating infectious agent. In this case, the individual is going to take more time to recover. This treatment take time, hence it can affect our daily activities. So if an individual is taking more time in recovering, then it's going to affect our daily activities. An infectious disease may transform from person who is suffering the disease to the another meanwhile of the treatment. If someone who is treating the infected person has a chance of suffering the same disease, So these are the limitations of treating infectious agent via medicines. Therefore, we should find out a way to prevent these diseases in the first place. And there are two ways, general method and the specific method. In general method, whatever is the source of infection is, for example, if the disease is waterborne disease, if it's an airborne disease, vector-borne disease, these, the sources has to stop. Water, in case of waterborne disease, have safe and pure drinking water. In case of airborne disease, avoid overcrowded places and keep the environment clean. And if it's a vector-borne disease, it could be prevented by keeping our surrounding clean and maintaining a public hygiene. So these are the general way in which we can prevent exposure to the infectious agent. But then there is a specific way which strengthen the immune system so that if any infectious agent enter our body, it can fight back. This can be made possible by having healthy food and through vaccination. Now the term comes, what is an immune system? It's a system of network of different cells, tissues and organs that work together in order to protect our body from diseases. We may consider the immune system as a defense system of our body. The immune system look out and destroy the disease-causing germs in our body with the help of special cells, and those cells are white blood cells. These cells are present in blood and hence circulate throughout the body and monitor it. The germs or any foreign substance that enter our body are called antigens. As the immune system recognizes these antigens, it releases antibody. So any foreign particle is known as antigen. Here, in this case, these viruses are antigen in this image, and the immune system is going to produce chemicals, and those are antibody, which lock the antigen and hence destroy the antigens. The ability of a body to resist a disease with the help of these antibodies is known as immunity. Here you can see on the screen different immune organs are there. Bone marrow that play an important role in production of B cells, spleen, thymus, Peyer's patches, tonsils, lymph nodes, appendix. These all are the part of immune system. Now let's learn about the discovery of Helicobacter pylori, a bacteria that was a cause of ulcer formation in stomach. A peptic ulcer, peptic word indicates the stomach, pepsin. A peptic ulcer is a sore that occurs on the inner lining of our stomach. In 2005, Barry Marshall and Robin Warren received a Nobel Prize for discovering this Helicobacter pylori bacteria as a cause of such ulcers. Before that, it was believed that stress and the lifestyles are the major cause of such ulcers in our stomach. The Helicobacter pylori bacteria weakens the mucous membrane that is there in the stomach, which leads to which lets the acid which is present in our stomach get inside to the stomach to get to the inner sensitive lining and damage it, and that leads to the formation of ulcer in the stomach. The acid and the bacteria together irritate the lining and cause a sore or ulcer in our stomach. So before 2005, before this discovery of Helicobacter pylori, it was believed that the stress and the lifestyle are the major cause of such ulcer in our stomach. But later it was discovered that there is a bacteria which is Helicobacter pylori, which is responsible for peptic ulcer. The specific method of strengthening the immune system is vaccination. The methods that we discussed till now were the general. general way just prevent the disease, right? But specific method strengthen our immune system, hence they provide, preventing ourselves against disease, and that specific method is vaccination. Whenever our body is affected by disease, our immune system not only fight against it, but it also remembers how to respond when the same disease causing microbe affect our body the next time. Similarly, vaccination contain an agent that is similar to the disease causing agent. For example, of the virus, a particular, if this is a structure of a virus that has protein, that is RNA inside it, a particular piece of that virus, a particular piece of the virus is inoculated, injected inside the healthy person, that will give the immune response. So here, vaccination contain an agent that is similar to the disease causing agent, which is weak or killed microbes. As it enters in our body, our immune system learns to fight against it and hence prevent us from actually getting infected when the actual disease causing microbes enter in our body. Today, vaccines are available for enhancing our immune system against various diseases such as polio, chickenpox, and measles. On our screen, we have different diseases, the causative agent, mode of transmission, how it can be controlled, and prevention. Malaria, the causative agent is Plasmodium. It is transmitted through bite of female Anopheles. It is controlled through quinine, which is a drug, medicine. It can be prevented by breaking contact between female Anopheles and man or eliminating Anopheles. Diarrhea is a protozoal disease. It could be viral disease. It could also be a bacterial disease. And it transmits through food and water. So it's a foodborne disease or waterborne disease. How it can be prevented? It can be prevented through proper sanitation, proper personal hygiene. Then comes the cholera, which is the causative agent is a bacteria. The name of bacteria is Vibrio cholerae. Typhoid, the causative agent is Salmonella typhi, which is a bacterial disease. Tuberculosis, against a bacterial disease. The causative agent is Mycobacterium tuberculosis. Hepatitis is viral disease, and there are around hepatitis virus from category A to G. So they have different hepatitis virus A, B, C, D, and so on. Then comes the disease rabies, which is vector-borne disease, and it is a viral disorder. AIDS, viral disease, and the causative agent is human immunodeficiency virus. Lastly, influenza, which is also a viral disease. The causative agent is myxovirus. Now let's know about Edward Jenner, which is known as the father of immunology. Why he was known as father of immunology? Because he was responsible. He is the one who discovered vaccine. During the late 1760s, he was serving his apprenticeship as a surgeon. Edward Jenner learned of the story common in rural areas that dairy workers would never have the often fatal or disfiguring disease like smallpox because they already had the cowpox, which has a very mild effect in humans. He gave the term vaccine in 1796, and by using cowpox, the Latin name of vaccinia is adapted, that means a cow. And vaccinia, that was from the cow, he inoculated into the humans. The cowpox from that lady, he inoculated into the humans, and he provided the protection against smallpox. Let's discuss the entire event, how he discovered vaccine. Edward Jenner was a good scientist and he really wanted to conquer the deadly smallpox. One day he observed that Sarah Nell, an ordinary milkmaid, get infected by cowpox? Nothing. The common observation that milkmaid were generally immune to smallpox. Jenner suggested that the pus in the blisters that milkmaid received from cowpox protected them from smallpox. So, Sarah was once having the cowpox. So, when she comes in contact with the smallpox virus, she was not affected. She got immune. So Jenner tested his hypothesis by inoculating the blister, the pus from Sarah, inoculated into a boy named James Phibbs, an 8-year-old boy. He scraped the pus from cowpox blisters on the hands of Sarah Nels. After inoculating, he subsequently observed that in James, fever and some uneasiness occurs, but no full-blown infections occur or observed. Then later, he injected Phibbs with variolous material, that is active virus of smallpox, but no disease followed in James. The boy was later challenged with variolous material and again showed no sign of infection. So eventually, vaccination was accepted, and in 1840, the British government banned vaccination, the use of smallpox to induce immunity, and provided vaccination using cowpox free of charge. The entire event at a glance, Sarah Nims, a milkmaid which was infected with the cowpox. James Fish is inoculated with cowpox pus from nuns. So the pus was inoculated in James. James fall ill but with a mild case, unlike smallpox. So he was having mild symptoms. Then scabs were collected from a smallpox patient and was inoculated into James. And James showed no symptoms at all. He remains unaffected and protection is complete. So this is how the concept of vaccination comes, that when mild or attenuated viruses or bacteria is inoculated in human body, our body get immunity against the deadly diseases. So this is how the concept of vaccination and immunization comes. Thanks for watching.